going a little faster than I normally do, so bear with me. And what we don't finish today, I'm going to have to transfer to tomorrow. But I do want you to have the material on hand. So what are the learning objectives in regards to periodontal bacteria and the immunology of periodontal diseases? One has to do with the concept that we're talking about a disease or a series of diseases that are not caused or don't have an etiology related to one bug, one bacteria. So I call this the concept of one bug, one disease versus mixed infection. So you may have learned about the famous 19th century microbiologist by the name of Koch from Denmark, I think he was, who was famous for having identified, and this is 19th century, late 19th century, when people couldn't even put their hands around the concept of a bacterium, let alone associating it with the disease. So you may have heard of Mycobacterium tuberculosis, and the principles related to the isolation of a certain organism and the principles behind isolating that organism from a human, putting it into an animal, and recapitulating the disease in that animal as it appears in a human, and then isolating that organism from the animal that is essentially the same organism that was isolated from the human. So those are really three principles related to Koch's postulates associated with one bug, one disease. Unfortunately, periodontal disease or periodontal diseases is not a one bug, one disease situation. It has to do with a mixed infection. So you've got multiple bugs, and the Academy of Periodontology changes almost like the weather as to whether the disease is one or it's many, and then they have subtypes, and we'll talk about that in brief. But this is something that I want to impress upon you, and also that the periodontal pathogens, you hear the word pathogen, are they really true pathogens? Is a periodontal organism that's really part of the commensal microflora of the mouth that under opportunistic conditions shifts into a microflora that could be detrimental to our periodontium, is that a true pathogen? Salmonella typhimurium is a true pathogen, right? Organisms that cause cholera are true pathogens. Mycobacterium tuberculosis, that's a true pathogen. Okay, these are organisms that shouldn't be in us, but when they do get in us, they can cause disease. These are organisms that are in us. I could do PCR analysis of everyone's plaque in this room, and I bet you everybody's got Porphyromonas gingivalis to some degree. Does that mean you have the disease? Not necessarily. So these are concepts that you need to understand, and we'll get into when we talk about dysbiosis. Specific versus non-specific plaque hypothesis. I don't know if have you already had lectures in Perio by Dr. Basada or any of the other? Did they talk about the concept of specific versus non-specific plaque hypothesis? Did that concept come up? No? All right, so we'll touch upon this. It's essentially saying, in different ways, what is the disease caused by? Is it a bug-only disease, meaning the bug is there, the bug goes up in high numbers, the bug causes the disease? Or is it a host-related disease? Doesn't matter what bug is there. Doesn't matter what bug is there. It's the way the host responds to that bug, and that can be genetically predetermined. There's something called, back in the late 90s, there was some really neat work demonstrating that there are people who are hyper-inflammatory phenotypes, meaning they've identified single nucleotide polymorphism, SNPs, in genes related to cytokines. And those SNPs, if they appear, are essentially causing overexpression of those cytokines in that individual. So the baseline levels of cytokines in that individual is elevated above other people. So you can conceive of the fact that these people could be at higher risk for periodontal disease. And that essentially is the connection. So is there a genetic predisposition to periodontal disease associated with commensal bacteria? But this has been ongoing for close to 100 years. Specific plaque, meaning there's specific organisms that are causing this disease, irrespective of the host response. Non-specific plaque says to you, it doesn't matter what the commensal bacterial load is. It is the load. It means it has to do with amount of commensal bacteria and the host. So we'll touch upon that a little bit. I just want to bring it to your attention. Then we'll talk about Agrobacter actinomycetum comitans. It used to be called Acinetobacter, Actinobacillus. I'm sorry, Acinetobacter is another organism. Actinobacillus actinomycetum comitans, AA for short. We'll call it AA for short. All right? Now the reason I'm emphasizing these particular types of organisms, more so than the microbiologist who's going to talk to you about bacteria, is because that individual, who's a very nice person, bacteriologist, microbiologist, doesn't have I don't think the sensitivity for the importance of these organisms the way we do in the dental arena. So that's why I wanted to emphasize periodontal microbiology for you. So anyway, this organism, AA, has been strongly implicated in a particular kind of periodontitis referred to as juvenile periodontitis, or juvenile aggressive periodontitis. Have you heard of that term? You haven't heard about that. Okay, that's in the pedo arena, so we'll talk about that organism. Then we'll talk about the Bacteroides groups. So the Bacteroides organisms, it's an umbrella term for organisms associated with periodontitis. Porphyromonas gingivalis used to be called Bacteroides gingivalis. Tenurella forsythia used to be called Bacteroides forsythus. And forsythus or forsythia is named after the place where it was first identified, the Forsyth Institute in Boston. Prevotella intermedia used to be called Bacteroides intermedius. They all have Bacteroides-like characteristics, but because we've become a lot more sophisticated in terms of understanding metabolic byproducts, unique for these different types of species of organisms, they've separated them into these separate species. However, they belong to this umbrella group, Bacteroideae, the genus, and I can't even pronounce it. So look up the genus for Bacteroides. So those are the three organisms, species of organisms, very important in periodontal disease. Bacteroid DTs or something like that. Treponema denticola, or denticola, is a spirochete, and that's an organism worth mentioning because it has also been implicated in advanced stages of periodontal disease. A spirochete. Have you heard of syphilis? Okay, so the famous organism associated with syphilis is Treponema pallidum. Okay, and so this is a spirochete related to that family of organisms, but unique to the oral cavity. Then we'll touch upon what's referred to as the red complex, and this comes from work that was done by Sig Sokransky's group at the Forsyth Institute. He and Ann Tanner, Ann Haffagey. Ann Haffagey and Sigmund Sokransky were pioneers in the 20th century. He is probably considered the most influential translational microbiologist in the area of oral diseases. So he passed away a couple of years ago, but he established this thing called the red complex, and we'll touch upon that as well, because they did a lot of prospective studies in understanding how the disease progresses longitudinally in people and the organisms that emerge as the disease is progressing. A lot of really very difficult, a lot of incredible hard work, big teams of people, a lot of studies on a great number of subjects, and to their credit, they established this thing called the red complex. And in the red complex, which is probably the three most important organisms in the exacerbation of the disease. So they determined through this process that Orphomonas gingivalis, Panurella forsythia, and Treponema denticola are the three organisms in the red complex that exacerbate the disease when they're present. So we'll touch upon that. And finally, what I wanted to really emphasize is the notion of keystone pathogens and how in the oral cavity that leads to dysbiosis. Okay. Yes?
Now, are you saying the red complex are normal inhabitants of the microflora?
Yeah.
Or in non-normal?
No?
Just out of balance?
They out of balance.
Okay.
That's the whole, the enigma here. Do we call these organisms really pathogens? I mean, they do have an armamentarium of pathogenic properties. But when they're at low numbers, and the vast majority of organisms are gram-positive, fermentative type of organisms, the lactobacilli, streptococcal, actinomyces type organisms, these are kept at bay. And when the gingivitis that leads to early stage periodontitis happens, the microenvironment changes and there's a shift. And that shift promotes overgrowth of these types of organisms.
These three are all primarily anaerobic?
Yes, they're all anaerobic.
So until they get deep into the sulcus, they're controlled merely by
Well,
breathing.
they start to emerge even in the gingivitis situation. All right, but if left unabated, you know, that oral hygiene doesn't improve, it can get worse. So we'll emphasize that. Louis Pasteur, okay? Very famous French microbiologist, again from the 19th century. I love history, and I think it's extremely important to know where we came from. So he said there are some bacteria that cause a disease, but there are some diseases that bring about a condition that is ideal for the growth of some bacteria. All right, what does that mean? And it's incredible that this is 19th century, where, as I said before, most people didn't even know, couldn't even conceive of bacteria. And he comes out with this kind of statement. And it's a true statement. What does it mean? What's the first part of the sentence? All right, so that one bug, one disease, right? What's the second part of the sentence?
Well, it's sort of chicken and egg. But we'll get that.
So you got a disease caused by one bacteria. Then you got another kind of disease that brings about a condition, meaning possibly the microenvironment, a change in the flora, right, that makes other bacteria grow well because it's conducive for their growth. But they have nothing to do with the disease. Right? Some diseases that bring about a condition that is ideal for the growth of some bacteria. Now, they could or could not have something to do with the disease. Okay? So you got a disease maybe caused by a particular organism. And we don't know what that organism is. But that organism shifts the community of bacteria to a community that under normal conditions would not cause disease. But in this condition where the microenvironment is conducive for their growth, for example, oxygen tension. Some of these are anaerobes. They love to thrive in low oxygen tension. So let's say the disease promotes a low oxygen environment. These guys are going to start growing. They start growing, We isolate them, we find that they have virulence factors. Now these virulence factors manifest themselves, but only after or during the time when this microenvironment shifts. Does this make sense? Okay, so this is Louis Pasteur not knowing anything about periodontal disease, and is probably saying the most important concept that we're now grappling with in the 21st century. So what are periodontal diseases? It's considered a local infection by indigenous plaque bacteria. My big question is, are these true pathogens? I don't believe they are. My bias is they're opportunistic. Just like Candida albicans, 50-some% of humankind carries Candida fungus in their mouth. Do they have candidal disease? No. Under healthy conditions? No. Immunocompromisation is a different story, right? But if you're either immunocompromised or you overprescribe antibiotics, they're not taken correctly, you got a shift in the microflora, Candida can grow. Well, in this case, that microenvironment may shift, whereby these organisms start growing. The microenvironment is conducive for their growth, and these guys, and they're anaerobes, and they've got an armamentarium of proteolytic enzymes, they start manifesting with destruction of the periodontium. So I believe they're what referred to as opportunistic bacteria. And I mentioned it's a time-dependent local immune response that mimics the natural history of inflammation. It includes remodeling of subjacent connective tissues, including bone. And down here, it says tissue remodeling involves cycles of destruction and reconstruction. So periodontal disease is not static. It's very active, but it has periods when it is destructive and periods when it's trying desperately to reconstruct itself. So there are ups and downs in the disease. There are quiescent moments and active moments of destruction. And we don't know when that's happening. And what normally happens in publications, attempts to associate bacteria with the disease, is that people will take samples from humans in the pockets, but they have no idea as to what stage of the disease they're taking the samples from. Is it an active destruction? Is it quiescent? What effect does that have on the microbiota? An opportunistic infection reflects a failure of innate immune mechanisms. So when you have an opportunistic infection, that's telling you that the innate response isn't effective in eradicating that infection. Okay? So it shifts from a failure on the part of the innate response, and then you get this acute phase of the adaptive immune response, and you get an inflammation. The chronic immune response results in increase of specific defenses, so you start seeing humoral immunity come in. The latter part of this whole phase is when humoral immunity kicks in. The early stages are innate immune defense, followed by cell-mediated immunity, and eventually the humoral immune response kicks in, and so you get high titers of antibodies. Yeah?
What's the difference between humoral immunity and adaptive immunity?
Humoral immunity is an arm of adaptive immunity. Right? Humoral immunity is an arm of adaptive immunity. So adaptive, I like to call it the umbrella. I always call these terms, there's an umbrella term, and then underneath it, so you've got adaptive immunity where cell-mediated and humoral immunity come into play. What I'm trying to do is make this situation that can be simplified. I'm bringing out the complexity in the story. Reasons for uncertainty and confusion defining microbial periopathogens. Periodontal disease could be periodontal diseases. There's more than one form of periodontal disease. It can be aggressive, it can be chronic, it can be refractory, it can be juvenile, it can be generalized, it can be localized, but they're all due to mixed infections. And how many species of bacteria have been identified in the oral cavity? We now estimate over 700. Okay? Many of them are fastidious. They cannot be isolated clinically or microbiologically the way we normally isolate organisms, which means they can't be studied for their virulence factors. Some can, and those are the ones we're going to be talking about. As I said, many are difficult to grow. They're highly fastidious. We don't know all the nutrients required for their growth. Time of sampling may be wrong. I told you about the cyclicality of the disease. So if an organism is there in order to promote destruction, and you're taking a sample at a time when it's quiescent, when the disease is quiescent, you could be missing out on that bug. That's just one example. Different sites in the same patient may have different bacteria. And that's been found over and over again. We don't have all the same bacterial community in all the sites. Opportunistic species grow as a result of disease rather than as cause. That was the thing Pasteur said over 100 years ago. So there are what are referred to association studies, where they associate a particular organism with the disease. Okay? That's clearly not causality. It's just association. Remember the second part of Pasteur's statement. And then there's something called carrier states, where phenotypically the individual appears normal. Okay? Which means there is no disease, and yet the person is infected, i.e. the presence of a particular periodontal pathogen is in that site, and yet there's no disease. And the organisms themselves, like Porphyromonas gingivalis, not all PGs are created equal. That's why I say strains of putative pathogens may vary in virulence. Okay, some strains are more virulent than other strains. Some strains are more virulent because they've been infected with a bacteriophage. And when we talk about AA, I'll mention the fact that something called a serotype B AA was shown to be infected by bacteriophage that led to expression of a very important virulence factor that only serotype B has at high levels. So, with all of this confusion, in comes Sigmund Sokransky, and he creates his postulates, or his criteria for identifying organisms that are periopathogens. Okay, just like Koch did in regards to tuberculosis that I told you. This guy has, I think, 6 criteria that we're going to talk about in order to identify whether an organism is a periodontal pathogen. Well, clearly association is there. It has to be found in high numbers in proximity to the disease site. And it has to be absent or present in far lower numbers after clinical intervention. Subjects, meaning after treatment, well, that's elimination. I'm sorry, I'm jumping that's the last one. So it's got to be found in high numbers in the disease site, and it has to be absent or present in much smaller numbers in the healthy sites or in people without the disease. Elimination, that's where I'm getting at. Clinical improvement following treatment has to eliminate the putative pathogen from the periodontal lesion. Now, by qPCR analysis, that may not be true. You're not going to eliminate the organism because PCR is very, very sensitive. All right? He was saying this at a time when we didn't have PCR. We were depending on isolating the organism using clinical microbiological techniques. But clearly, going from a very high concentration to a very low concentration with concomitant reduction in clinical presentation is what they're talking about. Post-response. So the host has to respond to this organism. The organism must have high levels of serum, salivary, and gingival, when I say organism, I mean the host. Must have high levels of serum, salivary, and gingival crevicular fluid antibodies against it in periodontal disease patients. So what you do is, have you heard of gingival crevicular fluid? Does that concept exist? Okay, so in the disease process, this is really serum exudate. It comes from serum and it percolates through the periodontium and it gets into the pocket. If you isolate that fluid, you will see a huge amount of really neat stuff, some of which are antibodies, others are PMNs, residual products of PMNs. And as the disease gets more serious and the pocket gets deeper, you get a lot more of this crevicular fluid. So the host responds to these organisms by generating antibodies. So you expect to see higher titers of these antibodies against this organism, against a particular organism. Virulence factors. So you want to be able to isolate the organism and dissect out its virulence factors. Okay? So it must be found to produce virulence factors in vitro, which can be correlated with clinical histopathology. What that says is, organism X, bacteria X, you isolate it. Now, histopathologically, what do you see? Well, you're seeing collagen fibers break down. You're seeing elastin break down. You're seeing a lot of ground substance in the periodontium break down. You're seeing death of cells, right? You're seeing inflammation. So what the idea here is you isolate the organism and you start looking at, can this organism in vitro do the kinds of things that you're seeing histopathologically in the disease? Can it break down collagen? Can it break down elastin? Can it be inflammatory? Does it have an LPS that's effective in promoting inflammation? That's what that's about. And finally, animal studies. You want to be able to take the organism and put it in the right model of another animal, and you want to recapitulate the disease in that animal. Experimental implantation of the organism, in this case organism means the bug. I should have changed that. I don't want to confuse you. The organism must have high levels of serum, meaning that's the host when I say organism there, right? And here I mean experimental implantation of the bacterium into the gingival crevice of an appropriate animal model should lead to development of some characteristics of naturally occurring periodontal disease. So there was one very important paper in Science in 1987, I think it was 87, where some periodontists got together, some noted guys and gals, and they took a particular animal model, Macaca fascicularis, which is a monkey, and what they did was they ligaturized with nylon ligatures certain teeth in the mouths of these monkeys. Now, why do you think the ligatures were put in? In order to get periodontal disease, what do you first have to have? Yeah, but what's that called in a sense? Huh? No. Inflammation. What sort of preliminary inflammation is it? Gingivitis. All right? Yeah. So you got to have gingivitis before you can't have periodontitis without gingivitis. Never heard of it. You can't jump into direct periodontitis without gingivitis. That's telling you that that microenvironment, that shift, is extremely important. All right? So they tried to put these bugs into healthy sulci in the monkeys. They couldn't get the disease to take shape. They could only get the disease to form and to worsen if they first created gingivitis. So these ligatures were put around the teeth in order to generate localized gingivitis. And then what did they do? They took Porphyromonas gingivalis, at the time Bacteroides gingival. And they implanted it into these inflamed gingival pockets. Okay? And over a 40-day period, they followed the disease progression and it mimicked what is happening in chronic periodontitis in humans. A lot was done in studying the histopathology of the disease in those animals. They did it earlier in beagle dogs. They found a similar situation. So, animal studies: association, elimination, post-response, virulence factors, and animal studies. If an organism fulfills these criteria, it falls under the category of a periopathogen. And so, this is a table that indicates to you on the left names of microbes, on the right periodontal diseases. This is an old table. Some of the purist periodontists from the academy will say this is no longer valid, meaning in terms of up-to-date naming. I'm not here to tell you whether this is correct or not. What I'm telling you is that using the criteria of Sokransky's, they were able to identify certain bacteria associated with certain forms of periodontal disease. Rapidly progressing periodontal disease, adult periodontitis. Notice actinomyces viscosus in adult gingivitis. Okay? So that's a gram-positive organism. Okay? Now, today, I wouldn't go so far as to say if actinomyces viscosus is there at high levels, you're going to have gingivitis. It's a lot more complex than that. But if you look at AA, localized juvenile periodontitis, that is definitive. And that's why I'm going to stress that particular organism. Okay, a little bit about the specific infections and specific plaque hypothesis. So there's been a long-going debate in the 20th century, it continues to today, about the specific versus non-specific plaque hypothesis. And what they mean about a specific plaque hypothesis is that the commensal microflora that makes up that plaque is unique. It's like a signature microflora that's associated with periodontal disease. However, as I said, you can have periodontal sites in a particular mouth of an individual that has one microflora and another site of periodontal disease, a localized site in the same mouth, that doesn't have the same microflora, meaning there could be elements of that microflora missing in one of the sites, and still you have disease. Okay, so where is the specificity? You also have to remember that we all possess these bacteria. As I said, they're really part of the commensal microflora. Not everyone with elevated levels of these bacteria exhibit periodontal disease. So that means there has to be a host element, a host part of that formula that isn't being considered. It has to do with resistance, tolerance. It has to do, and I mentioned to you before, this hyperinflammatory phenotype that was discovered in the late 90s that may be associated with refractory periodontitis because these individuals are inherently producing relatively high levels of TNF-alpha, interleukin-1 alpha, and possibly beta, because the genes themselves have been identified to have certain single nucleotide polymorphisms in their promoter sites, so that when those promoter sites are activated, the levels of the products go up really, really high. So that's very, very important. And host factors must be important in periodontal pathogenesis. So all of a sudden, these guys come up and demonstrate that indeed the host is important in this. And this is a paper worth reading by Barthold and Van Dyke, Periodontitis, a host-mediated disruption of microbial homeostasis, unlearning learned concepts, bringing back the notion that the host is really important in this process. And by the way, Tom Van Dyke is a graduate of our school. He's a noted periodontist at the Forsyth, in Boston. He was, and I was really supporting his candidacy. He was going to be our dean, but his wife nixed Cleveland. We got a bad rap. Cleveland's got a bad rap. Here are radiographs. So this is just to demonstrate to you, from a radiographic perspective, what juvenile periodontitis is like, right, in a young person. What you're seeing here, have you seen juvenile periodontitis at all? No? Okay. So notice, first molars, look at that area right there. Look at that area in the incisors. Look at that area in the upper molars. What do you see? Say that? Bone resorption. I mean, this is really severe in a young person. But look at the localization of the disease. It's only hitting these teeth. Look at this. It's only hitting either the first molars or the incisors. And that is unique to juvenile periodontitis. Okay? Now there's a lot of theory as to why that is happening. And I'm not convinced about what is being written in the literature about why it is. There's some stuff coming out that came out of the University of Pennsylvania about the window of opportunity for an organism like AA to land in these particular areas, unique in the oral cavity of young children. And you can see this also in the deciduous teeth of these children, which means that the molars, the primary deciduous molars and deciduous incisors of these kids are also at risk. So there are attempts to identify these kids and to treat them with antibiotics specifically to get rid of AA. Okay? So this window of opportunity talk about, again, I don't believe it, so I'm not going to tell you what is. I urge you to look into it. It's really interesting reading. So there's this window of opportunity around age 4 or 5 when this organism is trying to emerge, and it circumvents the humoral immune system, and the host generates antibodies that are not effective in eradicating the organism. It's that kind of story. Yeah.
For this sort of severity, what does that look for as far as retention and eruption from the teeth?
What effect does it have? It has horrific effects on those same teeth.
Right, so long term.
They're going to lose their first molars, and they're going to lose their incisors, the central incisors, if you don't intervene. There are some Japanese periodontists who are coming out with regimens of antibiotic use in the children when these particular diseases are first identified. Right? They do a heavy dose of a lot of antibiotics against this organism, and they claim success. So the degree of destruction of the periodontium in those children goes down so that they retain their deciduous teeth long enough for the permanents to emerge, and the permanents are less likely to undergo this kind of destruction with that regimen of antibiotics. So you see it here as well. What did I say? Here's generalized aggressive periodontitis, also associated with AA, where you see more than just one site or those specific sites that I mentioned before. Generalized aggressive periodontitis, the rate of attachment loss and bone loss is rapid in general, in generalized aggressive periodontitis compared to chronic periodontitis. That is true. So the rate of bone loss that one follows in chronic periodontitis I think I've got a slide that talks about how much bone is lost over time in a chronic periodontitis versus an aggressive periodontitis. It's unbelievable. Now, adult, this is not, what I'm referring to here is more juvenile, but the adult generalized aggressive periodontitis is not really associated with AA as much as it is with the host response and with those red complex organisms. So it's more than just AA alone. And the reason being that the AA that's associated with the juvenile form burns out after the patient has reached about 20 or 22 years of age. So you first see it, the serotype B that we'll talk about, emerge in these children. You follow these kids, you treat them, and then when they become adults, there's no longer this aggressive periodontitis. But in adults who develop generalized aggressive periodontitis, there's a host response element along with other bacteria that are involved in the red complex. So just distinguish those two conditions. Okay. A little bit about Agrigobacter actinomycetum comitans. Oh my God, I'm running really far behind. Gram-negative rod, doesn't form spores, non-motile, it doesn't move, doesn't have flagella or anything like that. Facultatively anaerobic, which essentially says it can grow in microaerophilic to obligate anaerobic conditions, so the oxygen tension doesn't need to be severe for it to grow. Forget that. Normal member of the oral flora in about 36% of the population. Now that depends on where. In the US, maybe that's the number. In Japan, it could be different. It's isolated primarily from subgingival plaque. It could be found in supragingival plaque and on certain mucosal surfaces. Okay? Remember, in the case of gingivitis, these organisms start to emerge, even though a gingivitis condition is aerobic, is still aerobic. All right? But there are micro areas within the gingival pocket where these organisms will predominate and start growing a lot because of the microenvironment being conducive. Catalase positive. Now, why is that important? I mentioned to you in innate immunity salivary peroxidase. Go back. Know about salivary peroxidase. It's a catalase. Now, why is that important? Gram-positive bacteria produce H2O2, hydrogen peroxide. What is hydrogen peroxide known to do? Aerate an environment. To kill off those obligate anaerobes. Right? So what does this guy do? It's got a catalase. It can catalyze hydrogen peroxide into water. So even though the gram-positives are producing H2O2 to destroy or to attack the AAs of the world, right? This guy produces a catalase that offsets that hydrogen peroxide. All right? So what I'm getting at is there are strategies that organisms use and have evolved in order to offset strategies of other organisms that are there to kill it. Obviously gram-negative, they're rods. Where's it isolated from? You can isolate it from actinomycotic lesions. Now this is a misnomer because it sounds like mycotic means fungal, right? But it isn't really a fungal infection. It's an infection with actinomyces species of organisms. Because when they first identified it, it looked like hyphae, and hyphae is that form of Candida that makes it virulent. That's the virulent form of Candida. So they first saw it, they thought it was mycotic, they called it actinomycotic lesions. But in fact, it's really a bacterial lesion. So AA associated with this in mucosa can lead to actinomycotic lesions. What is the colony morphology? I'm just going to give you a little bit of overview. It's small, circular, convex, translucent colonies, star-like centers. I think I've got this. So this is, if you shine a light on it, it looks like a star. And these are the colonies on sheep red blood plates. Broth cultures exhibit granular clumping. If you isolate it in broth, they grow like this. This is very important, the serotypes. The A strain, serotype A, is frequently found in the oral cavity, but it has what is referred to as a variable leukotoxin. Now I'm talking about a very important virulence factor, leukotoxin. This is extremely important. The strain, the B strain, is the most frequent in localized juvenile periodontitis. It's the one that always expresses leukotoxin. Okay? The C strain has no expression of leukotoxin. And that's usually, those are the kinds of AAs that you'll find in adults or in children who are not affected by the disease. But the B strain, and this is the one that was identified at Columbia, followed by work at UPenn, to have been infected by a bacteriophage that conveyed the genomic material that expressed the leukotoxin. Really interesting type of stuff. In regards to colonization, it colonizes both the plaque and mucosal tissues. It's got fimbriae that bind epithelial cells well, interbacterial interaction. So Strep sanguinis, Actinomyces viscosus, Strep uberis, these are kinds of gram-positive bacteria that have been shown to inhibit the growth of AA. But AA produces a bacteriocin, which is its own antibiotic, that can inhibit the growth of these organisms. Okay, so again, there is the fight for survival in the oral cavity. Virulence factors, I can't emphasize more. Leukotoxin, I say here it's a heat labile toxin. What does that mean? What does heat labile mean?
At certain temperatures, it becomes inactive?
Exactly. Heat labile versus heat stabile. So LPS is got lipid in it. If you've got a bacterial product that has a lipid virulence factor and you heat it, it's going to still be active after you heat it. On the other hand, protein, okay, if you heat it, especially if you boil it, it's going to lose its activity. So one of the ways to determine whether a bacterial toxin, a bacterial virulence factor, is protein or non-protein is to heat it. If it loses its activity, it's going to be proteinaceous. And that's what this is. But it's been shown to kill PMNs and monocytes. I don't want to go into immunosuppressive factors. The thing that I'm most interested in your knowing is this leukotoxin. Okay? It has a unique ability to kill PMNs. It also has some activity against complement, but primarily it will kill those cells that are coming in as the first line of defense, and therefore permit its growth. And this is just a cartoon to demonstrate the fact that what is happening is that you've got bacteria that are promoting let's say a gingivitis, or you've got bacteria that are promoting an epithelial response to the bacteria. So cytokines are being released, right? Tomorrow we'll talk about what's going on in the blood vessels that promotes the unique chemotaxis of PMNs from the blood vessels to the site. So there are receptors that we'll talk about tomorrow that are on these white blood cells. Okay? And they will bind to things on the surface of the endothelial cell. These things are called selectin molecules. Did Dr. Levine tell you about selectin molecules? Okay, so I'm going to reiterate it as it relates to periodontal disease. And eventually that slows down and it binds to ICAM, intercellular adhesion molecule. Okay, and that causes an anchoring of the white blood cell. And then there's diapedesis that's caused because of a chemokine gradient that's coming from here, right? Like IL-8. So essentially these PMNs are coming over here. But then all of a sudden, if there is serotype B, AA, releasing that leukotoxin, it's going to kill these guys. All right? And then you're not going to have the kind of defense you need in order to eradicate the bacteria. Simple, but important for you guys to know. This is really interesting, and this is something we have to be aware of. This is a whole gamish of AA by electron microscopy. But notice these guys that are coming out. Notice the blebbing off of the surface of this one. Notice that, okay? Those are microvesicles released by the bug, which have expressed on them the same kinds of things that are on the membrane of the bug. And they're highly inflammatory. So you can have a situation where the bug is in the crevice, and the bug is not motile. It doesn't penetrate the tissue, okay? But it releases these vesicles. And when it releases these vesicles, the vesicles themselves can percolate into the tissue. And they can promote inflammation. All right? So this is an important point that I want to stress, is that these organisms have the ability to promote inflammation even if they themselves are not getting into the tissue. Okay, how much time do we have? Not good. All right, B. frag, Porphyromonas, Prevotella, Penurella forsythia. All right, let's move fast on this. So this is what it looks like in a gram stain. They're gram-negative rods. This is the genus Bacteroides. They're strict anaerobes. That's not all that true. They can grow in low oxygen tension. They are non-motile, just like AA. They don't have flagellum. They do not form spores. They colonize the human mucous membrane. Now, there are both fermentative types and non-fermentative types. This is an interesting story because I've been telling you that the gram-positives are always fermentative and the gram-negatives are chemoorganotropic. They produce a lot of enzymes. They break down amino acids for energy. Well, it turns out that there's certain gram-negatives that also ferment sugars, and there are a number that don't ferment sugars. The other thing that they do is they sequester iron from red blood cells. Okay, that's called pigmentation. Iron is black. So these guys will grow on red agar plates, blood red agar plates. This is usually sheep red agar. And as they grow, they go from white to a dark blackish color. That means they are sequestering iron. They're really destroying the red blood cells and sequestering the iron for their growth. So there are types that are pigmented and those that are not, meaning that that will sequester iron, and then there are those that don't sequester iron. Anyway, what I'm getting at is the fact that I don't want to go into the intestine, but B. frag is something that is unique. It is probably the most important bug in the gut after E. coli in complications relating to surgeries. If you don't prescribe the right kind of antibiotics, B. frag comes up and becomes an opportunistic pathogen you have to deal with. This is a non-pigmented saccharolytic, which means it does not sequester iron, and it ferments sugars. Look at Tenorella forsythia, it used to be called Bacteroides bursitis. Look at Bacteroides intermedius, that's what it used to be called. Now it's Prevotella intermedia. And where is here? Porphyromonas gingivalis, used to be called Bacteroides gingivalis. Notice that Tenorella forsythia does not sequester iron, and it is asaccharolytic. Notice, I'm not done yet. You can come back in 5 minutes. Unless you want to sit here and learn something.
I could work.
Okay, sit down, sit down.
But I left my computer out in the hall, so I'll be back. Oh, yeah, when Dr. Weinberg's done, take a 10-minute break and then we'll start, okay? Thank you.
Okay. Where was I? Bacteroides intermedius. Prevotella intermedia, okay? Black pigmented, but fermenting. It's a fermentative anaerobic bacteria. Porphyromonas gingivalis, black pigmented, asaccharolytic. So this is one that you got to worry about because this guy does not ferment sugars. This guy has a huge armamentarium of enzymes. Okay? This is really a nasty bug. And I'm going to have to stop here, unfortunately, and we will continue tomorrow. All right? So tomorrow what I want to do is finish up on this and then give you an introduction on the importance of neutrophils in periodontal disease. And if there's time, well, tomorrow is at 3, so if you bear with me a few more minutes beyond 4, we can finish this up. We can have a menu review on these materials. Okay, see you. Bye-bye.
Thank you.